We're very lucky to have Sergei Kosakovsky speaking to us today. He's especially well known for his work developing methods in molecular evolution focused on the study of viruses, and for developing the Hi-Fi software package for likelihood-based testing of phylogenetic hypotheses. He's currently an assistant professor at the UCSD School of Medicine. His talk today is entitled Accurate Estimation of Evolutionary Attributes of Coding Sequences and Pattern Analysis in Large Collections of Viral and Mammalian Genes. Take it away, Sergei.
All right. Thank you for inviting me, Eric. I haven't done any web seminars before, so hopefully that'll go well. Usually people look away from the camera, so I'll try to actually look at it and pace myself reasonably well. I'll tell you a little bit about model development and sequence analysis work that we have been doing over the last couple of years, and we finally published 2 papers on it that sort of summarizes all the development, and I'll try to give you a little bit of a framework for how we came to think about molecular evolution in these particular terms and what types of problems we're trying to tackle. People that study sequences in a comparative framework have been using evolutionary models since Joe Felsenstein introduced, well, actually before Joe Felsenstein, but he popularized it in the early 1980s. And since then, a variety of models have been developed to estimate biologically interesting parameters from sequence data, things like evolutionary rate, divergence times, selection signatures, and the latter is something that we have been focusing on quite a bit, using this famous quote that all models are wrong, but some models are useful. What I mean by that is as computational power is becoming increasingly commoditized, you have multi-core machines, GPU machines, and I'm pretty sure that Mark Suchard talked about that when he gave his seminar in this series. Multiple distributed clustering environments under MPI, and a lot of data availability, specifically next generation sequencing, which for better or worse, we will all have to analyze almost exclusively, I would imagine, the next couple of years. With this, we can fit more complex and hopefully biologically realistic models to richer data sets. And I will explore some statistical and computational improvements in maximum likelihood-based inference from coding sequences and what they enable. If you haven't seen this before, I'll give you a very, very brief sort of conceptual overview of what a codon substitution model looks like. It's a Markov process, which, to be completely precise, is also time-reversible, stationary, and homogeneous. And what it's used for is it's used to parameterize probabilities of character substitutions in a phylogenetic tree that relate protein coding sequences. The structure of a Markov process is completely determined by its rate matrix, and the rate matrix tells you something about how we think the evolutionary process operates. So the rate matrix that I have on this slide, there are 3 components to non-trivial rates, and they come from the amino acid level substitution rates, nucleotide level substitution rates, and character frequencies. The nice thing about codon models is that they combine mutation and selection, and this is what made them so popular, specifically because they can be used to make inferences about various selective processes. Amino acid matrices have been around a lot longer, so Dayhoff is the model that we all know. BLOSUM is another matrix that's used extensively in BLAST protein sequence searches. But codon models have come to be sort of practically useful in the mid-1990s when computers became quick enough to actually compute something more than just 3 sequences using codon models. So initially in my talk, I'll focus on estimating the substitution rate term, which parameterizes how quickly 2 different codons that encode potentially different amino acids are exchanged. So this R term, which depends on the source amino acid, target amino acid, and potentially on which site in the alignment we're looking at and which branch in the tree we're looking at. So this is the most general possible parameterization. So if we were to take the most general parameterization just directly, and we wanted to fit this type of model to N sequences and S sites, the most general parameterization would have basically an enormous number of parameters that exceeds the most optimistic estimate of the number of available samples to us. And this is a statistically untenable situation. You want to have more samples than parameters, otherwise accurate inference is impossible. So our idea is to say that biological data are sparse So we can try to basically reduce the number of parameters, but using a data-driven model-based clustering and reduction approaches to group similar sites and similar rates together and have them share evolutionary rates, thus reducing the number of model parameters and overfitting. This is not a new idea. To put it in a historical context, as I've shown in this table, it's sort of chronological, with the original models of Musgod and Goldman-Yang in 1994. The way this original model approached dealing with the substitution term was very simple, and it was driven mostly by lack of data and lack of computing power. And the original model assumed that synonymous rates happen in some background neutral substitution, synonymous rates form the substitution background, so you could basically assume that their rate is relative 1, and non-synonymous rates were estimated using omega. There was no site-to-site rate variation, there was no branch-to-branch rate variation, and the initial application of these models was to test for gene-wide selection, so whether or not the average selective pressure measured by this omega ratio is different from 1. A significant advance to this framework was proposed by Nielsen and Yang in 1998, where they allowed site-to-site rate variation. Notice there is still no dependence of the substitution rate on the actual residues involved, but allowing different sites in a gene to have different selective pressures measured by this omega coefficient permitted site-by-site selection inference, which has been cited thousands of times and applied to basically every modern comparative study that involves protein coding genes. This was later expanded in 2002 to allow limited branch-to-branch rate variation. I, together with Spencer Muse, who's in North Carolina State University, have relaxed one of the modeling assumptions by assuming that synonymous rates, but permitting synonymous rates to vary from site to site. And this is sort of something that people have begun to realize in the last 5 years, that synonymous substitutions are not selectively neutral. There are all kinds of biological processes, starting from, say, secondary RNA structure in viruses, selection for expression, selection for codon usage bias, that actually make this rate vary from site to site. And in the last 5 years, there has been truly an explosion of various modifications to these codon models on both frequentist and Bayesian applications, too numerous to list. But typically these papers sort of proceed by saying, this is an unrealistic assumption, that we can try to relax it and see what that does to the model. A note on modeling assumptions is that sometimes it's actually worthwhile always to critically evaluate all types of assumptions made in early literature. For example, for people who have done this type of inference, codon model-based type of inference in the past, basically you open any paper that uses your standard MG or GY class models, and a part of this model must estimate empirical codon frequencies. Must estimate codon frequencies in the data set. And the classical way to do it is just to estimate position by position nucleotide frequencies, take their product, and approximate a codon frequency that way. I've actually discovered almost by accident that this estimator is biased, and it's been used exclusively throughout the literature, and it could potentially lead to incorrect substitution rate classification. It's easily corrected and should be done for all codon model applications. So this type of observation, that basically an assumption that has been made in 1994 and has never really properly been questioned, could be easily shown to be statistically biased, which is not a good situation to be in. And this is a simple modeling assumption that we can remove. A more complicated.
Could you give a just like one sentence description of what the bias is like? I guess it's just because it's kind of a homogeneous thing, and so things that are
So the basic, the very simple explanation for this bias is because traditional empirical frequency estimators forget to account for stop codons properly. So, and the way to think about it, there is a paper in PLOS One that describes it in painful detail, but the intuition is if you simulate under a codon model with a particular set of frequencies, infinite amounts of data, and then use this frequency-based estimator to recover the frequencies that the model had, you would get a biased estimate because you don't correct for stop codons.
So maybe that's also an answer to Joanna Silva's question, which is what is the magnitude of the bias in kind of ballpark terms?
It's a good question. So the bias, the way we discovered this bias was by trying to fit the next type of models that I'll actually tell you about. We're trying to classify different amino acid substitutions based on which residues are involved. And for these models, the bias can be severe, so we can basically significantly overcount the number of rates, and we can get biases in the order of up to 50%. But these biases usually affect substitution rates that involve individual residues. So for instance, if you take in a traditional codon framework, it's not going to affect very much branch lengths, and it's not going to affect very much overall omega ratios. What it's going to affect is the estimate of, say, residue to residue rates, and that bias can be, you know, anything depending on the size of the data. We've seen it as high as 35, 40%. I can actually see the questions too, which is good. They're all in the same phyloseminar box, right?
Yeah, I mean, as long as you don't mind seeing the.
I don't mind.
Okay.
Well, you should probably draw my attention to them. So
Okay.
what we're really after is trying to model rate-residue dependence. And as I mentioned before, since the early 1960s, people that worked at protein evolution have realized that patterns and exchangeabilities differ between residues, genes, organisms, and genome types. And this little plot right here just illustrates how different substitution rates can be, where these big bubbles of things like isoleucine to valine are happening very frequently, and something that's quite radical like histidine, to a serine, or actually histine to a tyrosine is even worse. So all the realistic protein evolutionary models have accounted for it. And the traditional approach is to take a large collection of aligned protein sequences and train your model. You can do it by counting, you can do it by maximum likelihood, but fundamentally you end up with a numerical model that says the rate of substitution from this residue to this residue is, say, 0.3. If we wanted to approach this more from the standpoint of people that have looked at nucleotide evolutionary model, where you look evolutionary models where you try to find the structure of the model, which means, say, transitions and transversions happen at different rates, and we'll estimate those rates. It's very difficult to do in the protein space because the model space that we could consider is combinatorially large. It has more than 10 to the power of 250 models and clearly cannot be explored exhaustively. The model selection tools for protein and coding data that exist, so ProTest, which is due to David Posada and his group, and Bayesian model selection, which has been explored by John Holzenbeck, they have largely been limited to screening a small subset of such empirical models or their mixtures. So what's shown here in this little blob is sort of the statistics of various data sets that have been screened through DataMonkey, and they give you just a ranking, a relative ranking of models. So JTT is a good average model, WAG is a good overall models. But the point to take home, there is no dominating model that explains everything. There's quite a bit of variability depending on which organism you look at.
So that's kind of cool. So that's literally like all these people are submitting their jobs to DataMonkey, and DataMonkey is picking models, and then you just kind of take the average of the models that people have submitted on DataMonkey?
Yeah, so there's a stats page that does it for nucleotide models. It's not a scientific sample. It's like, you know, your CNN sample, which says, are you going to watch the World Series, that kind of thing. But I find it curious. You can do, so if you go to DataMonkey, there's a stats tab that gives you these types of statistics for all the data that have been uploaded. So at least with protein models, there is some model selection structure. With codon models, there is nothing like it. And this is what we were trying to address in our original research. So model space in codon models is still combinatorially large, so we can still not compare all possible codon models. There really is no adopted analog of protein empirical models. I'll mention something a little bit later that has been done. And typically, that's because it's difficult to train because you have saturation and divergent alignments. It's expensive. You need a lot of data. And for 15 years, most widely used codon models have basically ignored differing residue exchangeabilities and said the same omega ratio to all non-synonymous substitutions. And to any sort of biologist that has looked at gene function and protein structure, that should be a serious affront because that is a very, very biologically unrealistic assumption. Omega is allowed to vary between branches and sites, but still does not depend on the residues involved in the most commonly used models. This unrealistic assumption is likely to bias some of the biological inferences, but we don't really know which ones. So I'll try to speak to that a little bit later. To set the stage for a model selection procedure, I would like to describe the type of models that we're willing to consider computationally. And this is the class of time-reversible Markov models. So if you've seen a model test in nucleotide space. This is basically exactly the analog, where you start with the simplest possible model, which is the codon analog of F81, and it's a single rate model in which the rate of substitution does not depend on the residues involved, except that whether or not they're synonymous or non-synonymous. This is the basic model. Synonymous rate of 1, non-synonymous rate of omega. The pros of these models are easy to fit and interpret. The cons is they're obviously ignore biological realities. We can go to the other extreme and say, let's just fit the codon equivalent of Rev that's reduced to only allow one-step nucleotide substitutions between pairs of residues. So if you look at all possible 190 distinct pairs of amino acid residues in the universal genetic code, 75 of them can be exchanged from one to the other using a single nucleotide substitution. That yields 75 possible rates that you can estimate. The pros is that you can, this is the most general model in the class. The cons, again, is computationally involved. It'll overfit the data, and it will be very difficult to generalize. So, and to speak a little bit to that, even in nucleotide space, you have to have a lot of data to truly support the reversible model. You can usually constrain a few of the parameters. So you can constrain, you can go to TN93, or you can go to something that is not quite Rev. You can, for instance, constrain AT and CT to be the same. Again, there's actually a stats page in DataMonkey for that shows that Rev is selected very, very rarely. Intermediate complexity approaches sort of fall in this burst of activity over the last 5 years, and they attempt to address this issue with using a single omega without being computationally or statistically unreasonable. And there are two major approaches that I've found in the literature. And the first one is empirical, which attempts to model what an average protein would evolve like in the codon space. The two papers that I would like to bring your attention to are the ones from Nick Goldman's group, where they fitted what's called an empirical codon model by basically taking all the protein families from the PANDA database, which they curate and used to maintain, but I think, unfortunately, they've run out of funding and this is no longer being obtained. And this is a direct analog of something like the Dayhoff or the JTT model for codons, where you take a very large training data set and you try to estimate what an average protein looks like. The problem with this type of approach is that there really no such thing as an average protein. There's a distribution of proteins, so this model will probably do reasonably well in the training data set, but we don't really know how it's going to do in any particular member, any particular protein that might not have come from this training data set, or even something that doesn't look like an average protein. Tal Pupko and his group in 2007 published an MBE paper using a slightly different approach, where they take the standard codon model and propose to overlay it with the amino acid substitution matrix, where basically, for instance, if you have an I to V substitution of a particular rate, you consider all possible codons that could that start at an I and go to a V, and you partition all these codon rates so that the cumulative rate matches what's in the JTT matrix for I to V. So this is, it combines amino acid protein models with mechanistic codon models. And lastly, there have been attempts to deal with basically expanding codon models to include more parameters, and I call them mechanistic or structural types of models. And one approach was proposed by the group out of the University of Missouri, CODON. They have two papers in MBE, where they say that omega, the substitution rate between two residues, is now modulated by a term that depends on how similar the two residues are. And the way they model the similarity is to take a linear combination of 5 independent amino acid physical and chemical properties, for instance, volume, charge, side chain, etc. Another approach was to take different models that were inferred, for instance, for variable loops, solvent-exposed residues, cool residues, and fit a phylogenetic mixture of such models. So this was done by people at Cornell a little bit earlier in 2005. Another illustration of why we really should sort of move away from the single omega model is it's very easy to demonstrate just how poorly this model fits. Another problem with the single rate model is that all the papers that I've just cited in the previous paper, in the previous page, the way they assess how well the new models do compared to the old ones is to take their model, fit it to the data, and compare it to the single rate model. If you can beat the single rate model, you have found improvement. So again, in nucleotide space, it's the analog of saying I can outperform JC69 or I can outperform F81. So in codon space, it's actually much worse than that, because we conducted this thought experiment, which I didn't actually think would turn out the way it did. But what we try to do is to say, let's take a data set, let's propose 100 random models where we say there are multiple substitution rates, but we don't really care how they're assigned. We just randomly drop them into the matrix. So we say, I2V is in class 1, F2P is in class 2, et cetera, et cetera, et cetera. And we do it with 2 or 5 rate classes. And if you look at the table here, this plot, we fitted 3 different data sets: small data set, intermediate data set, and large data set. And just look at this last column. So what this tells you is that for the first data set, out of 100 replicates, 96 random models significantly using likelihood ratio tests outperform the single rate model. You can do multiple test corrections and you still get 80. And as you give it more and more data, you basically beat the single rate model every single time. Right? So it really is remarkably bad. Another way to visualize this, and again, it's another sort of small note in PLOS One that we published a few months ago, is if you look at the same type of data visualized sort of on a scale where what's plotted on the x-axis is the log likelihood, the fit of this model. We don't really care at this point about parameters. It's just raw likelihood score. So models on the left are the ones that fit the worst. Models on the right are the ones that fit the best. As you would expect, single rate model is on the left. The general reversible model is on the right. Remember, we haven't corrected for the number of parameters yet. These dotted lines is what does this correction. So basically, any model that's to the right of this this dotted line is statistically indistinguishable from Rev, and any model that's on the left of this dotted line is statistically indistinguishable from single rate. And these other guys, so LCAP, ECM, are the 2 models that have been proposed by other researchers. So LCAP is this linear combination of amino acid properties mechanistic model, and ECM is this empirical model. So notice that basically what we have is, and this distribution is for the random models. So random models, as the previous table summarized, beat the single rate model, but the Rev model is still far, far away from the 2 empirical, the empirical and mechanistic model that people have proposed. So we haven't quite reached statistical saturation. So we can do better. We can add more parameters. We can make the models more realistic. Another interesting observation to make is that, say, the second RHODOPSIN data set, this empirical model, ECM, that came from a Pandit, is actually worse than the single rate model. And the reason for this is RHODOPSIN is sort of not in the training data set from which this model was derived. So sometimes putting an empirical model without really doing model testing to see whether or not it describes the evolution of a particular protein well could actually worsen your fit compared to the single rate model. So what we're after is going into this, basically going to the right of the dotted line to see if we can try to find a model that doesn't have a whole lot of parameters but fits as well, statistically speaking, as the Rev model. To summarize this, we posit that for practical data sets, we can only infer a few substitution rate classes. So we shouldn't be fitting the Rev model. It's an overkill and it's statistically noisy. So we'll go for something like maybe 2 to 8. And we don't fix this a priori, we actually infer it from the data. However, an important observation to make is that having even 2 substitution classes is fundamentally different from assuming a single non-synonymous rate, and eminently more biologically plausible. And another design feature that went into this model is that we didn't want to make any assumptions about how these residues clustered. We wanted the data to guide us, basically infer this type of clustering from the data. And to do this, we use a genetic algorithm to search the space of all possible allocations. Another way to think about it is in terms of curve fitting, which is a classic. If you've done any sort of machine learning or model selection, then you know that there's a sweet spot between models that underfit and models that overfit. So if you look at this plot, which displays for a particular data set, in this case, group M HIV1 polymerase sequences, all possible substitution rates from one amino acid to the other. So they're ranked in increasing order on the x-axis, and DNDS rate is ranked on the y-axis. So this purple line, which you may, hopefully colors come through right. So this purple line is the Rev model fit. So it's nice, jagged, and noisy, but it shows a significant spread of rates going all the way from 0 to something over 2. The red line is the single rate model, which clearly does not represent biological reality because we have variability between rates. This blue line is the random model, which kind of explains why it fits better. It's just not a line. Anything other than a line will do better. And this green step line is basically what we're after. We're trying to look for smoothing. We're trying to approximate the number of rates while basically capturing the overall shape of the rev curve. And you can see, in terms of log likelihood score, what we've done here is compared an individual model against the gold standard, which is in this case the rev model. So single rate loses by about 2,000 likelihood points. You add a random 5 parameters, you can recover about 62 of these likelihood points, which is statistically significant. But if you actually assign them properly and use 7 parameters, you can recover almost all of it. So you only lose to the GA by 46 likelihood points. So you don't need 75 parameters to get this likelihood score. You only need 7. And you can get most of the biological reality of how these rates are distributed using very few parameters. Why do we want to use a GA? Well, fundamentally, we don't really know anything about how the search space of the model is structured. We don't know if we change one rate a priori, if it's going to be a massive change to the likelihood or a small change to the likelihood. We don't know if there's a single peak, single model that has a good score. There are multiple islands. So it's very similar to the complicated search problem, like the search for the appropriate phylogenetic tree under the maximum likelihood criterion. And GAs have actually been successfully used for that. So Garley, which is a package written by Derek Zwickel, is a very good example of using a GA for complex search problem. We ourselves have used it in a variety of applications, which I'll name on this next page. But from the standpoint of an implementation, they're very easy to write, so they don't require specialized samplers or proposal schemes. It's basically a uniform black box that you just encode your problem in a way that the GA understands, feed it through the black box, get a solution out. What I really like about GAs is that they're trivial to parallelize. So alternative search approaches like MCMC, for instance, they're notoriously difficult to parallelize efficiently. But in this case, you basically, as many processors you have, you can use. They tend to converge quickly. We've used these GAs to do a variety of search problems for things like lineage-specific selection detection, RNA model selection, recombination detection. What we found in all of these applications is that previously the state of the art is to use a heuristic or some sort of an empirical limited set of models that people have considered. If you just allow the concept of searching for a well-fitting model, you can almost always outperform all other heuristic and mechanistic approaches, because we can't really anticipate in a complex biological system what will matter. Which 5 amino acid properties, for example, matter in terms of substitution rates. So what a GA does, it tries to minimize some sort of a fitness criterion. This is an example of a GA model selection run, which looks, again, very similar to if you've looked at, say, MCMC trace files. That looks very similar, except in this case, we're actually trying to lower an information criterion score that increases fitness. So we start at some random position here, the algorithm bobbles down, gets trapped in a local maximum, gets knocked out of a local maximum, then does a while to sample space of solutions around the maximum, and in the end it converges. It converges in about 10,000 individuals, so 10,000 different models. Now, each of these models can be fit on a different processor, not all 10,000 at a time, because there's a concept of a generation. So each of these, there's generation 0, then generation 1, then generation 2. But you can basically parallelize each generation. So you can easily use 100, 200 processors for this run very, very efficiently. Another nice analog, which is unfortunately not nearly as rigorous as you would get with MCMC, is the concept of a credible model. So there's some cutoff that you can compute and says that all the models that fall below this cutoff are basically credible given this data set. This is also better than using a single, this is infinitely better than using a single model for inference, because this allows you to include uncertainty that's inherent in such a complicated inference problem into your downstream analysis. So the way we measure goodness of fit is we actually played a lot with it, and I'll admit it to you up front that we basically decide that this is an appropriate criterion based on simulation. Fortunately for us, we can cite other papers from statistical science where such an embarrassing non-theoretical approach has been accepted. It's actually quite common for complex problems to tweak a penalty term in a likelihood-based criterion validated by simulation and say it works. It works in our case. We don't really know why, but it works. So what I will say, though, in our defense is that we've started with a standard BIC criterion, which has nice information theoretic and statistical foundations. And you'll recognize it as this -2 times the log likelihood. This is the goodness of fits component. And the penalty term. So all we've done is basically double the number of parameters. In the traditional BIC, the number of parameters is this P times the log of the sample size. We basically overcounted the number of parameters by a factor of 2. And the way I can explain this is that if you have a model, a CODON model with 5 rates, you obviously have 5 numerical parameters to estimate the rate themselves. But you also have this difficult to quantify concept of how these parameters are assigned to individual entries in the matrix. So this overcounting the number of parameters basically kind of fudges the factor that we're not including the parameter space, which is discrete, which is the way we assign individual rates to individual residues in the matrix. We validated it very extensively by simulation, and what we found is that it works really, really well. What's plotted here is that we've simulated very simple case 2 alignments of different length from 10,000 to 10 million codons to sort of approach asymptotics. And what we've computed here is the number of effective extra parameters that you get by fitting a 2-rate model to a model that was simulated under a single rate. So this is a false positive type simulation. We take a standard codon model, we simulate data under it, and we fit the GA, which attempts to allocate 2 rates. Find the best model with 2 rates that are allocated in an optimum way to different substitutions in the codon matrix. And we'll want our select model selection criterion to reject this model. So the cutoff here is the red line. And if you look later at the form of the information criterion that we selected, basically this is the penalty term that expresses the extra number of parameters that we can explain using the data. So in this case, we shouldn't be able to add any extra parameters. And sure enough, for basically 99 out of 100 replicates, we reject the 2-rate model as we should. But notice this is basically as low as we can go because there are a number of models that get very close. And it also doesn't seem to depend on the sample size, so it's a nice sort of stable behavior. Another case is to actually do power simulations when we simulate model with 2 rates, which is here. And in this case, the more data we give it, the more confident the criterion is that we have 2 rates. So it runs away. This is basically if you have, say, 10,000 codons, the effective number of parameters that you can explain by a 2-rate model is 1,000, which is significantly more than the 1 parameter that you're adding. But on the other hand, 3 parameters are still rejected in favor of 2. And again, it doesn't really depend on the sample size. So if you're interested in this, this paper is in PLOS ComBio. It's called Codon Test something. I don't remember what it's actually called. But you can always email me if you're interested. And it's all implemented in DataMonkey and in HiFi. So you can just, if you want to play with it, just go to DataMonkey, upload your data, and you can run all of the you can see how all of this works. Simulation summary, using a conservatively penalized BIC, GA model selection performs really well. So basically 0 rate of false positives with a slight tendency to overestimate the number of rate classes in some settings, mostly due to unobserved substitutions in the data. So sparse data, 0 estimated rates, and just a technical detail. Power behaves predictably. It increases with the alignment information content, which is measured by the size of the alignment and the divergence. And the more separate the simulated rates are, and you can basically get power of over 95% if you give it enough data. We are trying to figure out why this works, but I'm actually trying to get a postdoc that knows something about theoretical statistics to tell me why it works. Maybe I'll actually try to figure it out on my own. So as an example of what this actually does in terms of model fit is we've taken various models, including the single rate model, empirical models, and the LCAP model. So these guys have been proposed in the literature before. This is our GA selected model, and this is the REV model. And the bold face in each of the rows indicates the best model for a particular alignment. So to make a long story short, GA is the best model using BIC on 5 out of 7 training data sets and Revis in 2 out of 7. You might ask, why doesn't GA beat Rev in all 7 cases? Because we're training the model on this particular data. And the answer for this is because we're training the model under a more conservative criterion. So this is a standard BIC, which, remember, our modified BIC doubles the number of parameters. So if we were to retabulate this table in this modified BIC, would actually, the GA would win all the time. But we kind of bias the fields against it by using a standard information criterion. And even then, we're still able to, in 5 out of 7 cases, obtain a better fit than anything else. More importantly, GA beats the empirical generalist models in all cases. It beats a structural model for all except one case, which is one of these guys. And interestingly, and this brings me back to the point from a few slides ago, empirical models sometimes can actually perform worse than the single rate model. So you need to be careful about using model selection. Now, for the type of inference this enables is the first thing is that I promised you in the beginning that this model attempts to cluster different residues into how exchangeable they are. And exchangeable in this case means at what DNDS rate are 2 particular residues that are being substituted. So this is a partitioning of all possible 75 rates in this vertebrate RHODOPSIN alignment. If you follow positive selection detection literature, there's been this sort of controversial series of papers coming out of Masatoshi Nei's and group that used a paper that was published in PNAS on vertebrate vision, dim light vision proteins called rhodopsins, in which they basically tried to prove using one example where experimentally validated, well, actually, I'm not going to go into it too much, but if you're interested in where this data set comes from, I just search vertebrate rhodopsin, PNAS, and Nei, and you'll be treated to some very interesting exchanges between people that think that likelihood methods for positive selection work and those that don't. It's kind of like the current debate in the US between the Republicans and the Democrats that will never really agree, sort of fundamental, irreconcilable differences. So that aside, rate clustering allows you to basically say which substitution rates are the same. So here, this little collection of 3 residues, leucine, isoleucine, and valine, are the most exchangeable residues, which you would believe. They're all very similar residues. They're all in the same class. So this is actually an information-rich plot because the shape of each box means if it's a polar or charged residue, and the color of each box means whether or not it's a substitution in the same similarity class or not. So things from a red box to another red box are conservative, and they preserve basically everything. So the thing to take away from this is that first, there are a lot of rates that are inferred from this data set. So you have a non-synonymous rate, a DNDS rate going from 0.02 for the most of the residues all the way to 0.7. Now think about it for a second. If 1 is neutral, basically means that these 3 residues are effectively neutrally exchanged. So there's no, if you believe that omega approximates some sort of a fitness parameter, then these residues are more or less equally fit. Doesn't really matter how they're exchanged or which of them is present at a particular site. Another thing that we get out of this is because we have model average estimates, again, this is not based in a single model. This is based on a collection of models that can credibly explain the data. We can also obtain confidence intervals in how reliably we can say that, say, leucine and isoleucine are in the same class, or say, serine and alanine, et cetera, et cetera. So that by itself, I think, is really kind of interesting because that is a direct analog of what you would get for model test on nucleotide data. Now we have a model that partitions different residues into different bins. So now we can say that why are these substitutions different? So let's actually answer this question. So previous models have always assumed that substitution rates are driven by the desire to preserve a particular property. So the common wisdom is if you have a conservative residue substitution versus a radical residue substitution, the former will be more tolerated than the latter, and the former will happen at a higher rate than the latter. Right? Radical substitutions change the structure of the protein. My question is, do we know what constitutes radical? if we don't have the structure of the protein, if we don't really know what the evolutionary imperative is, very difficult to say a priori what constitutes a radical substitution, other than just it's a different amino acid. And one of the things we found is that if we take this model-based rate selection and compared it to what people would think is important, the answer is sometimes properties are important, sometimes they're not. We don't really know a priori which ones are. So in this RHODOPSIN data set, for instance, it is important for that data set to preserve similarity classes, and using a particular scale called Stanfield class, which takes all 20 amino acids and breaks them into 4 groups. Substitution within a group is conservative, substitution between groups is radical. So if you do this, then staying within the group is preferred. But polarity, for instance, this data set doesn't matter. Charge matters. And then we can also do, this is just categorical comparison, change or no change, low rate or high rate. And here what we're doing is comparing some, so for every 2 residues we can compare how similar they are on some chemical scale, for instance, chemical composition, polarity, or volume, and contrast this again against how rapidly they're exchanged. And you would imagine that the higher the difference between the residues, which is here, the lower the substitution rate. Sorry, so the substitution property-based distance increases along this axis. So things that have high property distances will also have lower rates. So you expect negative correlation between how different the residues are and how quickly they're exchanged, using this null hypothesis that proteins try to replace similar things with similar things. Now, what effect does this have on downstream inference? And it depends on what you're trying to infer. It has a very small effect on branch length, which basically is highlighted here. It doesn't really matter which model you use, single rate or the GA, you get more or less the same branch length. But it starts mattering a lot when you get into inference problems that really rely on the structure of the model. For instance, if you were to try to map substitutions on a phylogeny, you would get rather different answers depending on which model you use. This is a busy slide, and all I really want you to take away from this is that if I take the same site and the same alignment and just try to map where non-synonymous substitutions happened at this site, in this case I get 4 differences depending on which residue I look at. So it tells you that things that actually specifically ask about the probability of changing a given residue into another residue are significantly modified by allowing multiple substitution rates in the CODON model. And what does it do for detecting selected sites? So obviously, traditional tests just do D and DS. If average non-synonymous rate is greater than the average synonymous rate at a site and is statistically significant, we'll call it positively selected. But now we have multiple substitutions, so multiple tests can be carried out. The traditional test is DN greater than DS for every substitution class? We can also say, is DN greater than DS for at least some substitution class, or for a group of substitution classes? And to give you an idea of what this does, again, let's look at a RHODOPSIN data set. So that's just one site, CODON 54 in this data set, where if you use a traditional DNDS based selection detection method, you'll have a p-value of 0.03 that the site is under positive selection, positive diversifying selection. And you kind of can believe it, because if you look at the composition of amino acid residues and map all the substitutions, basically everything that happens at the site is a non-synonymous substitution. But you can also ask which non-synonymous substitutions contribute to it by testing individual substitutions with an individual class. And those classes were defined by the graphs a few slides ago. So in this particular case, substitutions are driven by class 1 and class 4, so they contribute to the overall positive selection signal. Another case is a site which is considerably more heterogeneous, so you have red non-synonymous substitutions, blue synonymous substitutions, and orange multiple substitutions. So if you run a positive selection test, standard positive selection test on this side, you get a p-value of 0.2, so it's not positively selected. However, if you actually drill down and start asking questions about individual sites, all of a sudden you have class 3, which actually pops out as positively selected. So the way to think about this is that now you actually have more power and potentially more noise and more false positives. We're actually investigating this, so don't quote me on that yet. But because you have more parameters and more resolution to ask questions about individual types of substitutions that are happening at a particular site, you can actually ask questions about which class of substitutions drives selection at this site. All right. In conclusion, so we've developed a method to rigorously classify amino acid substitutions into various exchangeability classes within evolutionary models. As far as we know, so we've really tried to find all the empirical and structural multi-rate models that people have proposed, and we've conducted a comparison of our models against best-in-class empirical and structural models, and our models tend to beat them. An interesting observation that I think is really striking is that a measurable proportion of substitutions evolve neutrally under selection on average in some genes, in which traditional DNDS measures less than 1. So if you go back to the 1990s when people were excited about the CODON models, but then they realized that basically every gene that you look at has an average DNDS of something like 0.1 or 0.2. So if you look at this level, positive selection operates in very few genes. Most things are conserved. Then the big breakthrough was to say, which parts of the gene are in a particular type of selection? What we're trying to say here is that even if you forget about site-to-site rate variation, you can, if you start asking questions about which types of substitutions are under selection, you can find in some strongly conserved genes, on average, some types of substitutions that are on average neutral or positively selected, like this isoleucine, valine, or leucine classes. Multiple class substitutions are now supported in addition to site-to-site variability and selection pressure. New models ideally can lead to better and more sensitive methods for detection of not only sites, but types of substitutions that are in positive selection. What do you want me to finish, Eric?
You got a couple minutes, but yeah, I mean.
All right. So, which sort of leads me to the next big application of this model. So, and I'll just briefly sketch it out, and I'll refer you to some other. If you're interested, you can read the papers and ask me questions. So a question about what size of data set. So basically, this data set, the model selection process itself is adaptive. Oh, you want to ask at the end?
I that's fine. Whatever you want to do. It seemed like you had kind of moved on, so.
Oh, no, this is a good question. So basically, the model selection procedure will adapt.
Wait, let's just read, because the people that are watching the recording can't see this text. Let me just read off the question. Joanna Silva asks,
what
size of data set is needed to get the necessary power to look at classes of substitution?
So what we've seen on practical size data sets, and practical size data sets, I mean something like more than 10 sequences up to a few hundred sequences, the more data you give it, the more resolution you get. And the philosophy behind this is that if you have infinite data, you will always select Rev. There is no biological reason to believe that any two substitutions happen at the same rate. Okay, so think of this as basically zooming in with greater and greater resolution as you have more data. So let me sort of give you a picture that might illustrate this. It's not really about rate selection, but it's the same type of selection problem where we try to cluster things into similarity groups. So here we're actually trying to cluster. As you can see, I've actually run out of time before I got through the second part of my talk, but that's okay. I'll just use this slide as a prop. So here on the left, it's a fixed size data set, and I'm trying to see, can I classify individual sites into classes based on their DN and DS rates. Okay, so with two-rate classes, I basically find this pattern first. So I find a pattern that there's a blob here that sits. So let me actually explain what this figure is. So on the x-axis, I have the log of the synonymous substitution rate. On the y-axis, I have the log of the non-synonymous substitution rate. This dotted line is DNDS equals 1. Everything below is negatively selected. Everything above is positively selected. Everything kind of on the line is neutral. So for this particular data set, I'm just basically trying to fit models with increasing levels of resolution. So I get first these two blobs, which tells me that the two predominant patterns in my data set is this almost neutral class and this negatively selected class. If I add more data, I can resolve this class. If I add yet more data, I can resolve this class. And you can think of this as basically the more data you get, the finer pictures you can resolve. But the original blob here and blob here persist as you add more and more data. But practically speaking, 10 sequences or more, you can't really do anything with fewer than 10. I mean, then you're just basically fitting the fact that most of the substitutions will be unobserved in these data. So let me conclude by saying here, I'll just sort of jump to this slide and talk for a few minutes, try to explain where we're all going with this. So what we have now is the ability to take an alignment, run it through a computational model selection procedure, come back with a model that says, how does this protein evolve? If you feed it two different data sets, you're guaranteed, given the number of the search of potential models, you'll end up with two different models, right? So give it HIV, I'll get one model. I'll give it, mammalian mitochondrial DNA, I'll get another model, which you can actually compare these models in different genetic codes. It's not a problem. You can just fit individual models in the right genetic code. So if I do this enough times, I can basically come up with an evolutionary description of a lot of different genes. And this is something we're trying to do with DataMonkey. We're actually going to add an option on it to allow people to basically add everything that's been inferred about their data set, but not the data set itself, to this database of evolutionary patterns. And we're getting, a few hundred jobs every day. So hopefully by this type of non-scientific sampling, we'll get nice coverage of what it is that people care about. And with this, each of these data sets, if people choose to run CODON model selection, they'll actually get a model that describes how this protein evolves, which by itself is useful. So you can then go and ask specific questions about this protein, estimate branch lengths, estimate substitution rates, estimate selective pressure, just look at which amino acids are similar. But then in the grand scheme of things, as this picture begins to fill out, so we have more and more data, you can start asking global questions, as in which types of substitution models do different proteins have? Does the fact that two proteins have similar functions, or they come from similar organisms, or they sort of sit close to each other on an evolutionary tree, does that imply that they have similar evolutionary models? So with this, we're basically trying to add another dimension onto evolutionary analyses. So you do what you usually do. You analyze basically everything you can out of an individual alignment, but then you can start asking questions. How does the evolutionary pressure of this alignment relate to other alignments? Can I find some long-range interactions? And just to give you a flavor, one of the things we've done is this type of analysis for different genes in HIV, for instance, so what we're doing now is fitting genes that are different. They're not homologous. You can't really compare them. You can't build a joint tree on a GAG and an RT and an envelope gene. They're different genes, but why are they sitting in the same tree, right? It's the same organism, different genes in the tree. So what we've done is basically a model-based clustering of these genes, where we've fitted a model to each gene, and compared it, devised an evolutionary distance metric between these two models, and constructed the similarity tree on the models. And our hypothesis was, do things with similar functions have similar models? And not surprisingly, they do. So in HIV, for example, all the polymerases are sitting together in this tree, or if you can visualize it in a non-parametric way, just principal component analysis, they're all together. GAG is by itself, ON by itself, and then multifunctional viral factors. The same is true for hepatitis C. The same is true for influenza. And more generally, if you start throwing a lot of alignments into this type of analysis, you can start asking sort of global, silly questions about, are there different types of evolutionary modes that proteins follow? And based on our initial screening of 650 different viral genes, there aren't. So these sort of psychedelic color plots, they look very much, these are nonlinear principal component analyses of these model components from different data sets. So what I'm analyzing here is 650 different alignments to each one of which we fitted CODON evolutionary models and compared the distance between these models for every two data sets. So it's conceptually similar to taking 650 sequences, comparing pairwise distances on them, and doing cluster analysis or neighbor joining tree, except our point now is a gene, and what we're building our distance based on is the model. So the fundamental advance here is that we can start comparing things that are actually not homologous. And you can start asking questions as, does evolutionary process differ, or is it similar between things with similar functions? So what these plots kind of shows is there's this whole spectrum in some abstract space where these models live that sort of populate. So one of the hypotheses you could have had is that there's sort of a bunch of fixed evolutionary modes. So you have genes that are strongly conserved. They sit in one own area of the space. You have genes that are under strong positive selection, so things that have to do with immune defense or adaptation. They sit in another part of an evolutionary space. But we actually have the entire spectrum, which reflects the fact that there is no such thing as a truly conserved gene. Parts of every gene is going to be under some sort of selective pressure, just like there's no such thing as a truly variable gene, because even in things like viral envelopes, there are motifs, secondary structures that are strongly conserved. So one of the things we have discovered is that there are some trends. So one of the trends that we expected to find, and we did find, is that there's broad separation between things that differ by function. So surface and capsid proteins in viruses and polymerase and transcription proteins in viruses, they kind of sit in different sides of this spectrum. I mean, they're not purely isolated. There are things in between, but overall there's a trend for this. We're trying to actually do this type of analysis on a whole genome scale with yeast and Drosophila to basically try to apply these ideas to clustering to find if there's association between evolutionary process and, say, protein interaction networks or gene ontology terms, or you name it. So the nice thing about it is that we actually have a way to measure the distance between genes using these types of evolutionary models. All right, so I would like to acknowledge all of my colleagues. This has been sort of a multi-year effort, including several institutions. Wayne Delport, a research scientist in my group here, Conrad Scheffler, a computer scientist in South Africa, Spencer Muse, my original co-author of Hi-Fi and a statistician, Mike Gravener, a medical researcher in Wales, Art Poon, sort of a former postdoc who's now moved on to greener pastures in Canada, your neighbor, Eric, and Simon Frost, who's at Cambridge, and all the funders, which in this case is mostly NIH and SF and various collections of HIV-specific groups. Well, thank you for your attention, and hopefully it wasn't too jumbled at the end. I apologize for running out of time.